Hi, my name is Madeline Mariani, and I am a senior at Columbia College studying neuroscience and behavior. And this summer, I worked at the Ultrani Lab studying hippocampal neurogenesis. One of the projects I'm working on is single-cell RNA-seq data analysis from post-mortem adult human hippocampal tissue. So first, I'd like to say a little bit about the experimental and data processing workflow. So single-cell RNA-seq gives us the ability to sort or cluster cells based on the genes they express, so we can classify them as more or less similar to their cell types and identify these clusters based on the markers. And then we had about 2,400 cells from 4 samples, and after quality control, we ended up with about 1,700 cells because we exclude cells that express low levels of RNA or very few genes because these cells may be dying. So once we cluster the cells, we can go through and look at the differential expression in these cells to find their identity. Here we have 10 clusters: oligodendrocytes, endothelial cells, astrocytes, microglia, epithelial cells, ependymal cells, oligodendrocyte progenitor cells, excitatory neurons, excitatory granule neurons, and immature neurons. Using known markers of neuronal cells, we're able to categorize our 3 neuronal clusters more specifically. So first, we can see that they all express RBFOX3, which is a known neuronal marker, and SYT1, which is a marker of synapses. And we can see that the excitatory clusters express it more highly than the immature clusters, which is expected. Then we can see that the granule neurons express FOX1, which is a known marker of granule cells, and that the excitatory clusters express this, which is a marker of glutamatergic signaling, and thus excitatory neurons. Finally, we can check out a known marker of immature neurons, DCX or double cortin, which is expressed most highly in immature neurons, and you can also see that they express GAD2, which is a marker of GABAergic neurons, which helps us to identify this cluster because we know that immature neurons are inhibitory. So here we successfully applied single-cell RNA-seq to postmortem human hippocampal tissue. And once we're able to collect some more data, hopefully we'll be able to see some new markers of these cells based on the known markers and identify more specific cell identities from these clusters. Thank you.